Introduction IB Bio 2.5 notes. First, I'm going to be talking about enzymes' active sites. Enzymes are large polypeptide chains with a tertiary and quaternary structure. Enzyme substrate specificity. Only one type of substrate fits into the active site of a specific enzyme. The active site is the result of the way in which the polypeptide chain folds. Enzyme changes shape slightly to accommodate substrate. This is called induced fit and is possible thanks to the flexibility of protein molecules. Enzymes weaken the bonds within the molecules of the substrate, thus reducing the activation energy needed for a reaction. Next, I'm going to be talking about a catalyst, the catalysis. Activation energy, minimum energy that reacting particles should possess in order for a reaction to occur. A catalytic reaction is when an enzyme converts a substance into products. Enzyme catalysis involves molecular motion and the collision of substrates with the active site. Endothermic reaction is a reaction in which energy is gained. Exothermic reaction is a reaction in which the energy is released. Next, I'm going to be talking about the rate of activity of enzymes. Temperature affects enzymes' activity. When temperature is low, molecules tend to move slowly, meaning less collisions and therefore less enzymatic activity. When temperature rises, molecules move more rapidly and collide more, meaning more enzymatic activity. Each enzyme has an optimum temperature in which the rate of enzymatic reaction is the highest. If the temperature is higher than optimal, an enzyme can be denatured. As a result, the rate of enzymatic reaction rapidly decreases. Substrate concentration. When the substrate concentration is low, there are more enzyme molecules available than substrate. The rate of reaction is relatively low because of this. Increasing the substrate concentration causes more chances of the collision between substrate and enzyme molecules. Hence, the rate of enzymatic reaction rises gradually. However, this increase is halted when all active sites are occupied by substrate molecules. pH. A change to pH from the optimum affects enzymes and their activity. Extreme pH values can denature an enzyme by altering the three-dimensional structure of its active site. If pH is lower or higher than the optimal, the rate of reaction gradually decreases. For the next slide, I'm going to be talking about use of enzyme in the industry. Enzymes are widely used in the industry, such as medicine, fuels, agriculture, and even production of textiles. In order to make enzymes more stable and useful, they need to be immobilized. Immobilization is a process of attaching an enzyme to a material so that its movements are restricted. If enzymes are not immobilized, they are often present in the final product, which restricts the concentration that can be used to process food for human consumption to avoid adverse effects. The use of immobilized enzymes permit higher concentrations of enzymes to be used, allowing a faster rate of reaction. This allows immediate separation of the enzymes from the reaction mixture, which allows them to be recycled, reducing production costs. For the final slide, I'm going to continue on the use of enzymes in the industry. Lactose is a naturally occurring disaccharide present in mammalian milk and dairy products, and made of glucose and galactose. Lactase is an enzyme that can break down lactose into monosaccharides. Lactose intolerant people lack lactase and can't digest lactose, as it remains in the digestive system and creates gastric problems. Advantages of lactose-free products. They allow them, they allow It allows immediate separations of the enzymes from the reaction mixture, which allows them to be recycled, reducing production costs. No ill effects after consumption, and quicker fermentation. For example, in yogurt production, as bacteria ferment glucose and galactose are readily than lactose. It's sweeter tasting milk. Glucose and galactose are sweeter tasting than lactose. Lactose-free products are produced by adding the enzyme lactase to milk. Lactase breaks down lactose into its constituent monomers, glucose and galactose. In an In an alternative method, lactase can be immobilized in alginate beads while the milk is allowed to flow past. With this method, no lactase ends up in the final dairy product, making it better for consumption.